Peace to everybody tuned in to the Master Warrior Clan YouTube channel. I am your brother, Sean P. Most of you know me by any Ret, Sean Calvani. He who brings back the lost shall lead. Getting started a little bit late. But like the show, share the show, tell a friend, we live. I do understand that I think that Sister Dr. Mahal may be live, so I don't expect to get a lot of people in here today. Before we get started, though, I do want to thank everybody for subscribing to the channel who have recently subscribed. If you are new to the channel, please make sure that you subscribe, hit the like button, click on the bell. Let's go, man. We're going to get it in. I don't expect a large audience for this right now. You know what I'm saying? Maybe some replays later on down the line. But what up, Teddy? I see you in the building, bro. What's good? Why I got you here before we really get it cracking, man. You got the book, right? I need somebody who got the book. I think you got the book. Hold, let me see. Peace of your hurry. What's good? What's good? Yeah, you got the book, right? Let me know. Anybody that actually got my book. I got a question I want to ask. And all right, okay. So have you read beyond the introduction or forward? Have you got into any other chapters? If so, which chapters have you read and which one have you learned the most from? So again, if you have the book right and read it, or whatever you've read thus far, which chapter have you read? If you've read multiple chapters, and which one have you learned the most from in the book? And for those of you who are listening in who may not have the book is in the description of the video. It's called What SARS-CoV-2 Taught Me. And if you hadn't got it, shame on you. Shame on you. Get your copy ASAP. Okay, it's chapter 4. Learn the most in chapter 4. Okay. Anyone else want to answer that question besides Teddy? Feel free to do so. 4 is informative. It's just basically showing you how COVID-19, how the disease works, how it affects us. Because our bodies have to go through a lot of damage. In some instances, when it's severe, in order to clear the virus, um, even asymptomatic people have to go through some level of sickness in order for them to clear the virus. So even if it's not severe and it's more, just the regular stuff, and I hate to say this, but we call regular stuff like sniffles and we, all of that. We call that just regular stuff, but those viruses have that effect on us, and it is what it is at that point. It's unfortunate, but that's the way that it actually happens. So I could see where you are with that one. Yeah. Chapter 2. Oh yeah. Feel you. I don't know if anybody else want to add to that before we get going, but uh-oh. Okay. Feel free again, man. Anybody joining us right now, like the show, share the show. We warming up a little bit before we get it cracking. We're about to get into it all the way. Um, and when we jump in, man, we're gonna jump in it and then we're gonna make it happen. So as you can see from the title, there's an emergence of a new vaccine. So if you do have my book, one of the things that I don't have to tell you or talk to you about is what is a vaccine and what is a vaccine's job, because in the book it guides us right down the path and understanding vaccines wholeheartedly. And you know, it is what it is from that point. So I don't want to go backwards to go forwards. I don't want to go backwards to go forward. So let's get into it. It's a big deal. It's a big deal. Like I said, this is gonna be a little light thing. I ain't expecting a lot of people to show up for this. You know what I mean? They might show up for the next show. I guarantee you. When I do the next show, they're gonna show up for that. It's a fact. So good catch. All right, let me share my screen, man. Let's get it going. So here we go. It's called Emergence Vaccines. Okay. And we're going to go through this pretty slowly because I want people to pretty much understand the logistics of this. Again, if you are unable to see this on your phone, what up, ATL? Change the quality on your cell phone from auto recommended to 720p, and your phone will clear up, and your phone will allow you to be able to see what I'm showing on the screen. And I'm gonna make it a little bit bigger if I can in some instances for people, and we're gonna get into it. So, okay, Emergence Vaccines' goal is to improve global health, human health, by providing long-term immunity for some of the world's most pressing diseases. That's the vision. So, what is Emergence? All right, what are Emergence vaccines? The Cowboys then scored again quickly. Emergence Vaccines is a clinical stage, privately held biotechnology company headquartered in Abingdon in the UK and focused on addressing some of the world's most immediate health threats, including viral diseases such as dengue fever, Zika, Ebola, and pandemic flu, as well as a series of intracellular bacterial infections. Emergex is developing a pipeline of innovative CD8 plus. If you're familiar with the immune system, you would know what CD8 is referring to, because it's finna tell you, ladies and gentlemen. CD8 plus, they didn't tie the game up. T cell priming vaccines that have the potential to deliver fast, broad, and long lasting immunity to reduce serious illnesses associated with infectious diseases. Now, Emergex vaccines are entirely synthetic without using a timeout. Again, Emergex's vaccines are entirely synthetic, which means that they do not require biological steps to be manufactured, and in turn do not introduce live viruses, RNA or DNA into people. An added benefit is that the synthetic vaccines can be stored at ambient temperatures, avoiding the need for refrigeration or freezing and making vaccinations much more convenient all around the world. Our T cell vaccine candidates have also been designed to be delivered transdermally, which means avoiding the need for traditional needles and intramuscular injections. Drawing on a rich scientific heritage and international collaborative approach, Emergex Vaccines have invested in state-of-the-art laboratories to conduct research and develop key components of its vaccines, and works closely alongside its international collaborators, including the Institute of Molecular and Cell Biology in Singapore, Biomimejados, Theo Cruz in Brazil, and George Mason University in the US. Now, the technologies as it goes, and then we'll take a look at the video. But they combine the validated technologies with the lasting scientific advances to develop our vaccines, including using synthetic peptide codes selected and determined through experiments on actual pathogen-infected cells and using their proprietary quantum size nanocluster carrier system for delivery of these codes. Our technologies are advantageous in numerous ways. They have a business model, and you can see this on the website. I'm not going to get into their business model. I'm more in tune to get into where we are with this vaccine. Hold up. Before I get him the ability to hit me with the umph. What up, Donnie? What's good, man? Peace to you. All right, so now we got that out of the way with the disclaimer. Let's proceed. It's gonna be a nice little bill because we're about to go in some directions y'all don't even think we're going.
From mosquito-borne viruses such as Zika and dengue, human-to-human transmitted viruses such as Ebola and pandemic influenza, to new strains of antibiotic-resistant bacteria, current infectious diseases, as well as emerging ones, continue to present a true challenge for our world. Viral outbreaks usually occur unexpectedly and spread rapidly. In the West African Ebola outbreak of 2015, there was no experimental vaccine available until the outbreak was well advanced. By then, the ring vaccination concept was not available in time to have a meaningful impact on mortality. The 2018 outbreak allowed for further testing of ring vaccination, but this does not address the issue of a vaccine for the general population to prevent future outbreaks. There are numerous high-risk diseases in terms of virulence. There are also newly emerging zoonotic viruses that present the potential for outbreaks in endemic regions. No effective vaccine exists for any of these infectious disease viruses. Current vaccine technologies are not suited to on-demand, real-time crises. Previous attempts at vaccine production have been costly in both development time and pricing. They contain live viral components and experience arduous development and manufacturing processes, which usually encounter production and reproduction bottlenecks, as well as lengthy timescales. The one bug, one drug approach of conventional vaccine development cannot conquer the battle against the multiple array of emerging infectious diseases. Emergence has developed a disruptive, novel approach to making vaccines that use purely synthetic components to activate specific immune cells that destroy virus-infected human cells. The components utilized to create the vaccines have already been proven safe in clinical trials. When someone is infected naturally by a virus, certain small proteins called peptides are expressed on the surface of the infected cells. These peptides are what the immune system targets and remembers to provide long-term immunity. We identify these peptides by infecting human cells with a virus. We then extract and sequence these peptides, which we use as the immunological components of our vaccines. These peptides, with a gold nanoparticle carrier system, create an extremely small construct that can directly enter immunizing cells. These then instruct the immune system to kill virus-producing cells, that is, the virus factory. Emergence's synthetic vaccines can provide protection against multiple diseases within a genus. For example, a single vaccine could provide protection against a viral family of more than 66 diseases. The development and production process is quick, inexpensive, and highly scalable. The vaccines are also stable in ambient temperatures, completely eliminating the requirement for cold storage and allowing for extensive shelf life. Emergex's vaccines will be administered via a microneedle skin patch. The patch will be designed to exactly penetrate the dermis. The vaccine will migrate directly into the lymphatic system, transporting the vaccine to lymph nodes for immunization. This simple and convenient delivery method means that there is less need for primary healthcare providers to be present for the administration of the vaccines. Patients can self-administer, thereby reducing the requirement for travel within infrastructurally challenged environments. Emergex's strategy is to advance vaccines against high-priority disease indications to Phase 1 human safety trials. We will then design and generate suitable repositories of experimental vaccines in readiness for outbreaks within endemic regions. Governments, multilateral and non-governmental organizations will either have direct access to Emergex's vaccines via repository prescription or will be granted manufacturing rights within that region. This provides a unique, on-demand, and real-time emergency line of defense against some of the world's most dangerous pathogens. Emergex is working directly with endemic region governments to provide national health services with the vaccines required to protect and/or treat these viral outbreaks. Emergex believes that it will change the face of the vaccine-producing world by creating smart vaccines by a cleverly crafted mechanism that reverse engineers the immune system and delivers data to relevant cells.
So there you have it. Emergex vaccines are on the way. The research and study is in process. Next thing you know, we'll see how effective and ineffective Emergex vaccines will be. I see that we're being joined by people who just showed up. So peace to everyone that just hopped into the chat. Basically, we were just dealing with Emergex vaccines, which are a new innovative vaccine that the way that these vaccines work are they're needleless, they're intramuscular injections. Is a delivering method that will be transitional as far as their approach, using their understanding of basically the immune system. So they're using the CD8 plus T cell priming vaccine. So basically, that's what they'll be. They'll be generated like that, and you have to catch pretty much the replay to get any further than that. Of course, they're hiring, right? They're looking for innovative ways to actually help people move further. And this T cell priming vaccine is evolutionary in a sense. It's just as evolutionary as mRNA technology, but it's different, right? And it's going to be what it's going to be. But Emergex are all about, look what it says. My bad. Again, if you can't see, change your quality in your settings to clear it up on your cell phone, and you should be able to see it. But Emergex has successfully generated a first generation human specific MHC class CD8 peptide linking them library for dengue, influenza, Zika, hepatitis B, and so forth. They are targeting these areas to fight, and it's going to get interesting. It's going to get interesting with this new vaccine, this peptide. So it's a combination of the technology that produces a set point vaccine capable of delivering the right peptides to the right place in order to produce a strong T cell immune response that will target and kill infected cells. So if you know anything about vaccine delivery systems or vaccine technology, go ahead and add this. This will be something that we'll continue to watch in its development to see if this is cool for us. It has a concentration in the areas of pretty much helping those in underclass worlds be able to defend and fight against the above mentioned viruses that plague some of the lower countries. That right there that I just highlighted. All right. So I wanted to start the show off with that before I move any further with the real good stuff. We about to get into some real good stuff. All right. What y'all think about that technology and its innovative ability to be able to target and attack, infected cells using peptides and being needleless? What y'all think about that? Anybody got any opinion about that before we get going on? Before we get started with the cream brulee, the dessert? None, nobody. Okay, cool. All right. Let me wait a little bit. I see we got people coming in. Like the show, share the show. It's about to get interesting. It's about to get interesting. Like the show, share the show. It's about to get interesting. So we're about to really, 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 really get the cooking right here. Really, really, really about to get the cooking. Now, if you have my book, this is why I ask people if you got the book. But if you have my book, this dude elbow is messed up. If you have my book, ladies and gentlemen, in the back of my book, there are 2 bonus chapters, right? One of the bonus chapters is on page 167. SARS-CoV-2 origins, bonus material. Let me see if I can do a little. First, let me drop the link to the book in the chat for people who do not have it. Man, get your copy of the book. And let me see if I can pull up the book. Hopefully I'll pull up the right copy, the edited version and not the unedited copy. I did a poor job in naming these things in my savings. Page 167. All right. I can show this. The reason that I can show this is because I will be revisiting this potentially in an up and coming text. Kenny, you late, bro. We didn't get busy. We didn't already talked about that new vaccine coming. We're gonna get it in. But if you have the book, you can go to page 167 in the actual book. This is the chapter on page 167. All right, it says SARS-CoV-2 origins bonus material. Come on now, like the show, share the show, tell a friend, tell a friend. I know most of y'all don't care nothing about COVID. You think it's fake. You think it can't. You think you're invincible. You think you're the most important organism on this planet. You think because you've caught COVID already that you ain't gonna catch it again. You have some type of protection. You think that it's all a hoax. You think that it didn't kill you. You could recover for it, so you don't need a vaccine. You think a whole lot of everything. I get it. I understand it. You're invincible. You're the smartest organism on this planet, and you don't believe that nature is really trying to kill you. You don't. You don't. I get it. All right, ladies and gentlemen, I get it. You think that you're smarter than nature. You think that you can defeat nature, and you think that all things natural are healthy. You think all things natural are good for you. You think all things natural are not an actual problem. 33 to 30. 33, 33. 19 seconds left. It's going overtime. Just to give you an update on where you are. There you go, and there you have it. Now, the origins of COVID are still being investigated. However, a few very shaky hypotheses has been regurgitated by non-scientists stating that the virus was man-made or potentially leaked from a lab. In chapter 1, I began with the viruses for a reason because I needed to make sure that you understand viruses are natural. Now, as we began to populate the globe, the more we became exposed to novel viruses. Currently, hold on, let me go back. Yeah, I knew some. Yeah. There we go. So I'm gonna run it to you again. The origins of COVID are still being investigated. However, a very shaky hypothesis has been regurgitated by non-scientists stating that the virus was man-made or potentially leaked from a lab. In chapter 1, I began with viruses for a reason because I needed to make sure that you understand viruses are natural. As we began to populate the globe, the more we became exposed to novel viruses. Currently, Cell.com has published a nice article on SARS-CoV-2 origins in bats. Results demonstrated a 96 to 97% match, providing a very strong case. However, we will continue to allow science an opportunity to have its say and not come to an absolute conclusion at this time. World Health Organization, was not convinced of the lab leak origins, but did not close the door on that option. The WHO study revealed two potential lineages of concern: the wet market in Wuhan and the domestic wildlife of some form. WHO stated tracebacks are very important in this stage. As for virus origins, if you think the Spanish flu evolved from Spain, that shows a lack of understanding of viruses on your part. Its origins have not been settled for over 100 years. Science has no consensus, however. Historians assume the origins are in China. That what many do not know about virus origins is that it is hard to narrow down a virus origin to a specific place. But after further research, we can potentially narrow it down to a geographical location. Swine flu was assumed to have its origins in the US, but later it was revealed that its true origins were in Mexico. There is much debate among virologists about this question. Three main hypotheses have been articulated. One, the progressive or escape hypothesis, states that the viruses arose from genetic elements to gain the ability to move between cells. Two, the regressive or reduction hypothesis asserts that the viruses are remnants of cellular organisms. And three, the virus first hypothesis states that the virus predate or co-evolved with the current cellular host. The progressive hypothesis, according to this hypothesis, versus originated through a progressive process. Now, mobile genetic elements, pieces of genetic material capable of moving within a genome, gain the ability to exit one cell and enter another. To conceptualize this transformation, let's examine the replication of retroviruses, the family of viruses to which HIV belongs to. Retroviruses have a single-stranded genome. And when the virus enters a host cell, a viral enzyme reverses transcriptase, converts the single-stranded RNA into double-stranded DNA. We know this because we're studying classes on virology, and in our virology class, not even a few weeks ago, we talked about transcription and where it goes from one phase to another. RNA can't be DNA. It has to be, because RNA is single stranded, it has to be double stranded in order for its conversion to take place. Now, the viral DNA then migrates to the nucleus of the host cell. Another viral enzyme integrates, insert into the newly formed viral DNA into the host cell genome. Viral genes can then be transcribed and translated. The host cell's RNA polymers can produce new copies of the virus single-stranded RNA genome, progeny virus is assembled and exit the cell to begin the process again. This process very closely mirrors the movement of the important, though somewhat unusual, component of most eukaryotic genomes, retrotransposons. Now, I'm talking over your head, I know. These mobile genetic elements make up an astonishing 42% of the human genome and can move with the genome via an RNA intermediate, like retroviruses. Certain classes are retrotransposons. The viral-like retrosponsones encode a reverse transcriptase and often an integrase. I'm going to stop right there because I don't want to muddy the waters up for you. And you'll have to slow down with me on this particular chapter because basically all I'm telling y'all, or all I'm doing right here is explaining how viruses transcribe from one phase to another, and is using different studies in order to obtain and get to that. All right. Now, not going to read this whole bonus material. I just wanted to bring it up on the screen because it's in my book. Get the book. Support the book. Read the book. Learn from the book. It's phases for a reason, ladies and gentlemen. Phases for a reason. Now, well, I just showed you how my book talks about, and let me put it back on the screen. SARS-CoV-2 origins is bonus material in the book. So let's get to cooking. All right. Let me pull the PDF up for y'all. So that we can look at this the proper way. I don't want this one. I need to map. Yeah, I need to. I think y'all should be able to see this, but let me see. Just hold on a second. Yeah. All right. So you should be able to see this, and I'm going to expand it out and I'm going to blow it up. And I want y'all to walk with me real quick on this. Oh, it's 165 in your book? Should be 167. Well, it depends on. All right. So I'm going to let the cat out the bag. It's 167 in the updated version of the book. If you got this book earlier on, I went back and I think I might have added something in there. Well, I took out a word or something, and then some kind of weird way something changed. But cool, no problem. 165, and some books is 167. It's okay. You got it. You got the info. All right. So can y'all see this pretty clearly? Do I need to zoom in a little bit more? I'm gonna zoom in a little bit more and let me know if you can see that clearly. If you're on an iPad, you may be good, but it's the people that's on a cell phone I'm trying to make sure can see this. Remember, if you can't see it good, to make sure that you change your quality because I can see it on my cell phone. Now I'm checking it. Okay. So again, so here we go. You should be able to either follow along with me if you know how to read it, if you can see it. I said if you know how to read it. And then we're going to talk about it. So dissecting the early COVID-19 cases in Wuhan. Elucidating the origins of a pandemic requires understanding of the Wuhan outbreak. Some key questions lie at the heart of investigation into the origin of COVID-19 pandemic, including what is known about the earliest COVID-19 cases in Wuhan, China, and what can be learned from them. Despite assertions to the contrary, it is now clear that live mammals susceptible to coronaviruses, including raccoon dogs, were sold at Hunan market and 3 other live animal markets in Wuhan before the pandemic. Severe acute respiratory syndrome related coronaviruses were found in raccoon dogs during the SARS-CoV outbreak, which was facilitated by an animal to human contact to live animal markets in China. However, because of the earlier public health focus of Hunan market, it remains unclear whether the apparent preponderance of hospitalized COVID-19 cases associated with this market was truly reflective of the initial outbreak. All right. Now, answering these questions requires resolving several crucial events that took place in December 2019 and early January 2020. On December 2019, the Wuhan Municipal Health Commission issued 2 emergency notices of internal inoculation to local hospitals, alerting them to patients with unexplained pneumonia. If you've listened to the video that I see, the video that I posted regarding my intro to the book, I talk about this, not in the same words, but I talk about this. Again, on December 30th, 2019, Wuhan Municipal Health Commission issued 2 emergency notices and circulation to local hospitals, alerting them to the patients with unexplained pneumonia, several of whom worked at the Hunan market and laying out a treatment and response plan. The first official public report was WHC's announcement the next day that the third had carried out a case searches and retrospective investigations related to Hunan market and found 27 patients. That number is significant. 41 of the first known patients from the basis of an influential study that reported that 66%, i.e., not all early cases, had a link to Hunan market. They had been transferred between the 29th of December and January the 2nd, and from other hospitals to Jintin Hospital. All right. Now, Wuhan's premier infectious disease center, notably individuals were enrolled according to clinical presentations, not epidemiological information, such as connections to Hunan market. Now, China viral pneumonia of unknown etiology mechanism was set up in a wake of SARS to be an early warning reporting system. All right. In this reporting system for detecting unknown viral diseases and is overseen by China Center Disease Control and Prevention. Vpu cases are supposed to be rapidly reported by clinicians to the National Notifiable Disease Reporting System through an internet-based platform. Evidently, that did not happen in Wuhan in December. The system appears to have been in active use only for from January the 3rd. Although it favored cases having a connection to a Hunan market, the Vpu mechanism could not have improperly inflated the proportion of Hunan market linked cases in December. Moreover, reporting began only after the 41 patients were transferred from other hospitals to Jintin hospitals. Nevertheless, it is possible that the disproportionate number of cases linked to Hunan were transferred to Jintin Hospital because of public health officials' early focus there. Now, there is, however, a way to step back to a period before any such bias could have crept in by considering what happened in the hospitals that first pieced together that a new viral outbreak was underway. Although not mentioned by name in scientific publications, media reports revealed that Vpu Principal Hospital of Integrated Chinese and Western Medicine was the first hospital to alert district, municipal, and provincial public health authorities about the mysterious pneumonia. Again, we talked about this. They had no idea what it was in its earlier onset in December. It was a mysterious pneumonia, unlike different pneumonias, and they didn't know. They had the first 27 cases, 27 turned into 41. They transferred these people to a different hospital. Now they are exposing people to something that they knew nothing about. Zhang insisted that a couple's son, who was not a patient and had no symptoms, undergo a CT scan, and the same unusual lesions were observed. The husband and the wife evidently are cluster 1. Here we go. We're not saying that they were the first people. Science is not saying that they was the first people, but we have cluster 1. Okay? The parents had something, and the child who they got a CT scan on had these unusual lesions. Now, cluster 1 in the World Health Organization China report. They are the earliest known case cluster and the only cluster admitted by December 26. They had no known connection to Hunan. So cluster 1 had no affiliation whatsoever with the market. Now, another patient with similar CT imaging, a worker at the Hunan market, was admitted on the 27th of December. Zhang, concerned about a new probably infectious viral disease, reported the 4 cases to the hospital officials, who alerted the Jinging district CDC at the same time. Over the 28th and 29th December, 3 more patients, all of whom worked at Hunan market, were admitted and recognized to have the same unknown respiratory disease. A vice president at that long-name hospital, Wingong, brought together 10 experts from the hospital, including Zhang, for an emergency meeting on the 29th of December. And they concluded that the situation was extraordinary. Upon learning similar patients also linked to Hunan market and Tong and Union hospitals, I don't know how to say that word, Qi alerted the Wuhan and Hubei CDCs on the 29th of December. This fool missed the field goal, but they called a timeout. He missed it. They called offsides again on the offense, on the defense again. That's the first down. Dumbass. All right, so a notably similar situation unfolded at Wuhan Central Hospital. On the December the 18th, a Fin director of the emergency department encountered her first unexplained pneumonia. Patient, 65 years old man who had become ill on either the 13th or the 15th of December. Unbeknownst to I, at the time, the patient was a delivery man at Hunan Market. A CT scan revealed infections in both lungs, and he did not respond to antibiotics or anti-influenza drugs. On the 24th of December, a bronchioliver lavage specimen collected from him was sent to Vision Medical's, a metagenomic sequencing company. They identified a new SARS-CoV. This happened on the 24th of December. So by the 26th of December, when this information was relayed, defined by telephone to the hospital on the 27th and the 28th, Wuhan had identified 7 cases, of which 4 turned out to be linked to the Hunan market. 7 cases, 4 linked to the market. 3 cases not linked to the market. Notably, these 7 cases, like those at that long hospital name, were ascertained by epidemiologic investigation concerning Hunan markets commenced on the 29th of December. At the Zone Hospital at the Wung district of Wuhan, 15 kilometers away from Hunan Market, and an opposite bank at the Yangtze River, Vice President Yu Feng as units on December the 31st to search for an unexplained pneumonia cases in respiratory medicine department reported 2. Different hospital, different location, 2 reports. I need y'all to understand what's going on here. Everybody kept saying the market, the market, the market. You had 7 cases, 4 Hunan market, 3 outside the market. Then you had people from different hospitals reporting cases. So they're tracing the virus. The second did not work at Hunan market, but had friends who did and who had visited his home. On the 3rd of January, 3 more cases was identified. A family cluster unlinked to Hunan market. Clearly, hospitals in the first weeks of the outbreak were identifying cases both with and without known connection to Hunan market. And Wuhan hospitals were not swamped with unexplained pneumonia cases at the end of December. That would come later. Thus, 10 of these hospitals, 19 earliest COVID-19 cases were leaked to Hunan market. Comparable both to Jenton's 66% of 41 cases for, and to the WHO China reports 33% of 168 retrospectively identified cases across December 2019. Regarding cases at Wuhan Central Hospital at that long name, patients with a history of exposure to Hunan market could not have been cherry-picked before anyone had identified the market as an epidemiologic risk factor. Hence, there was a genuine preponderance of early COVID-19 cases associated with the Wuhan market. How can this knowledge inform our understanding of the pandemic? Good question. How can, if Hunan market was the source, why were only 1 to 2/3 of early cases linked to the market? If it's the source. Perhaps a better question is, why would one expect all cases as ascertained weeks into an outbreak to be confined to the market? Given the high transmissibility of SARS-CoV-2 and the rate of asymptomatic spread, many symptomatic cases were inevitably soon lack a direct link to location of the pandemic's origin, and in some cases counted as unlinked, may have been only 1 or 2 transmissions away, as exemplified by the second patient identified at the Zone hospitals, that so many of the below 100 COVID-19 cases in December with no identified epidemiological link to Hunan markets nonetheless lived in its direct vicinity is notably and provides compelling evidence that community transmission started at the market. Like this show, share the show. We're dealing with some high-level science right here, because we're dealing with merely epidemiological studies, a compilation of a bunch of data. This information is not in my book, but it will add to the discussion. It will add to me going back and increasing the work in that bonus chapter. Additionally, the earliest known cases should not necessarily be expected to be the first infected or linked to Hunan market. They probably postdated the outbreak index case by a considerable period, because only 7% of SARS-CoV-2 infections led to hospitalizations. Most fly under the radar. Similar, it is entirely expected that early ascertained cases from seafood markets would be workers who were not necessarily directly associated with wildlife sales, because the outbreak spread from human to human. The index case was most likely one of the 93% who never required hospitalization, and indeed could have been any of the hundreds of workers who had even brief contact infected live and mammals. One of one. Crucially, however, the now famous earliest COVID-19 case, a 41-year-old male accountant who lived 30 kilometers south of Hunan Market and had no connection to it, illness onset reported on the 8th of December, appears to have become ill with COVID-19 considerably later. When he was interviewed, he reported that his COVID-19 symptoms started with a fever on the 16th of December. The 8th of December illness was a dental problem related to baby teeth retained in adulthood. This is corroborated by the hospital records in a scientific paper that reports his COVID-19 onset on the 16th of December and a date of hospitalization on the 22nd. This indicates that he was infected through community transmission after the virus had begun spreading in Hunan Market. He believed that he may have been infected in a hospital, presumably during his dental emergency or on the subway during his commute. He had also traveled north of Hunan Market shortly before his symptoms began. His symptom onset came after multiple cases in workers at Hunan Market making a female seafood vendor there the earliest known case with illness onset on December 11th. Notably, she reported knowledge of several possible COVID-19 cases in clinics and hospitals that were near Hunan Market on the 11th of December, and the Hunan Market patients were hospitalized at the Union Hospital as early as the 10th of December. Now, although a widely cited report credits the Vpu mechanism with uncovering the pandemic, it was the long hospital name that identified both the outbreak and the Hunan Market connection and passed only these fully formed discoveries to the district municipal and the provincial public health officials by the 29th of December. I hope y'all are following along with me because we're talking about the virus's origins. Now, I knew this would be a little tricky for some people and other people wouldn't be able to follow along. So I didn't expect a lot of people to show up for this. But we're talking about the virus origins and we're relating this back to a book. It's in my book. If you don't have my book, ladies and gentlemen, I don't know what you're missing because I'm giving you this data that is not in my book, previously reported. And now I'm giving you additional information because my book, I give you the 2 lineage lines of how this thing was sequenced out. And then I turn around and we're going to add to the discussion. And here we are. Although a widely cited report credits the Vpu mechanism with uncovering the pandemic, it was the long name of the outbreak of the Hunan Market connection and passed on these fully discovered to the districts. National officials reportedly did not learn about the outbreak until the CCDC director, Gaio, encountered online group chats about WHC emergency notices on the evening of the 30th of December. Concerned that so many cases had not been reported to the Vpu system, he quickly notified the National Health Commission. Therefore, the preponderance of early cases connected to Hunan Market could not have been an artifact of ascertainment bias introduced by case definitions in the Vpu system, although mechanisms like China's Vpu system are potentially invaluable. They will fail without both widespread buy-in from healthcare providers and rapid data sharing from local to central authorities. Key problems with the Vpu system were known before the pandemic, including that most clinicians in China had little awareness of the Vpu systems and were not reporting cases to it. For example, 0 of 335 Pew cases in one study from 2019. China could be commended, however, for having such a system, which is lacking in most countries. The focus now should be on fixing the problems that COVID-19 has exposed and blanketing the globe with a highly functional Pew early warning system. Samples from the earliest COVID-19 patients in Wuhan have been sequenced, and the 2 distinct SARS-CoV-2 lineages, A and B, have been identified. I'm going to get this to you again. If you got my book, then you've already known, and you heard me and Unc argue this several times, that there are 2 distinct SARS-CoV-2 lineages, lineage A and lineage B, have been identified. Given that the elderly couple at the Lone Hospital who was whose report cluster 1, it follows that the husband illness onset on the 26th of December must be the source of the earliest lineage A. That's key. Now, Wuhan IME WHO 2019 information ascertains, which he most likely got from his wife who became ill on the 15th. This raises the possibility that the Yukachu market, a different market that they visited, may have been a site of separate animal spillover. So it was not Wuhan. It was a different market site for lineage A or B, intermediates in humans. So let's just deal with it. Let's go. The recent discovery that there may be no true lineage A or B intermediates in humans also raises the possibility of separate spillovers of both lineages. However, the earliest known lineage of genomes have close geographical connections to Hunan market. One from a patient age and gender not reported who stayed in a hotel near um, the Hunan market in the days before his illness onset in December, and the others from the 62-year-old in the cluster 1 who visited the Yukachu market just a few blocks north of Hunan and lived just to the south. Therefore, if lineage A had a separate animal origin from lineage B, both most likely occurred in the Hunan market. And the association of the Yukachu market, which does not appear to have sold live animals, is likely due to community transmission. You keep hearing this word, community transmission, starting in the neighborhood surrounding the Hunan market. Now, with SARS-CoV-2 live animal markets continue to sell infected animals for many months, allowing the zoonotic spillover to be established as the origin of revealing multiple independent jumps from animals into humans. Unfortunately, no live mammal collected at the Hunan market or any other live animal market in Wuhan has been screened for SARS-CoV-2 related viruses. And Hunan market was closed and disinfected on January 1st, 2020. Nevertheless, that most early symptomatic cases were linked to Hunan, specifically to the western section where raccoon dogs were caged, provides strong evidence of a live animal market origin of the pandemic. This would explain the extraordinary preponderance of early COVID-19 cases at one of the handful of sites in Wuhan's population of 11 million that sell some of the same animals that brought us SARS. Although it may never be possible to recover related viruses from animals, if they were not sampled at the time of the emergence, conclusive evidence of Hunan market origin from infected wildlife may nonetheless be obtainable through analysis of spatial patterns of early cases from additional genomic data, including SARS-CoV-2 positive samples in the Hunan market, as well as through integration of additional epidemiological data. Preventing future pandemics depends on this effort. The full source is in the description of the link. Let me. All right, so here we have the COVID-19 cases in Wuhan in December 2019. This map shows that the earliest cases of COVID-19 were in close proximity to the Hunan market, even if they are not directly connected with the market through working there or visiting. This suggests that transmission in the community around the market was occurring in December 2019. The map is based on a subset of data from 174 COVID-19 cases in and around the Wuhan. Hold on one second. Y'all couldn't hear me at all, huh? All right. So in the red, we have home addresses of cases epidemiological link to Hunan market, and then no identified link to Hunan market in aqua. And then you have the circles is the market, the pluses is hospitals. All right. Remember, red, home addresses of cases with epidemiological link to the Hunan market. Aqua green, no identified link to the market. The black circles, market. The plus signs, hospitals. Hospital, 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 hospital. Link to the market, not linked to the market. Not linked, not linked, not linked, not linked to the market. Not linked, not linked. A whole bunch of not linked and some linked to the markets here. Hunan is here in the cluster, but around are people that are not linked to the market. Right? So you can see how they look way up here, not linked to the market. They found it way up here, way over here, way over here. If y'all can see my mouse, my bad, I'm acting like y'all can see my mouse. But to the far east, over 20 kilometers, this is like 20, this is like 50 kilometers away in either direction, 50 kilometers away or 60. This might be 60 kilometers away from Hunan. And this got to be at least 50, 40 here, potentially 60 here. These could both be 60 kilometers away. It's like 30. Chavis, you late. I've been talked about that. I'm an hour into this, bro. You late. It's in the first 22, 23 minutes of the video. Yeah, but see, this is where we have, this is, all right, so that's what you're saying, Shezmu, is solid. And I'm not going to argue with you on that. I'm actually with you on your thinking, Shezmu, because we got two lineage. We know for a fact that we got two lineages. We got lineage A and lineage B. That was reported by WHO, but that also shows up in other places, and I will show you that. All right, so I'll show you that in just a second. But the reason that I'm saying what I'm saying, though, Shezmu, is this. In my book, I specifically deal with there's levels to this. Conspiracists claim without any evidence that the people have modified and created ACE2, citing gain of function as a premise. When asked to explain gain of function, things begin to become murky, which is a sign that someone is regurgitating something someone else had said without providing any evidence. As mentioned previously, the study published in Cell uncovered 4 related novel coronaviruses found in bats, more specifically the rhinolapid bats, which were found in several different parts of Asia. Despite the discovery of animal coronavirus related to SARS-CoV-2, the evolutionary origins of the virus are elusive. We describe a meta-transcriptic study of 411 bat samples collected from a small geographical region in Yunnan province in China between May 2019 and November 2020. Jumping ahead, here we go. The genetic lineages of SARS-CoV-2 associated with the early cases in Wuhan are documented in the WHO report. Previously, Rumbach noted that at the root of the I don't know why I can't say this. Phylogeny of SARS-CoV-2, guys, Lee, I don't know what's up today, are two lineages designated lineages A and B. Early lineage A virus, SARS-CoV-2 isolated. They give us the sample, person linked to wildlife market, different than the Hunan market. We go there, and then we get into B, lineage B, such as SARS-CoV-2 isolated, Hunan, General Bank of Sessions, molecular epidemiology sample, and then it goes from there. So I give you the A, B in the argument here. I don't know why this is so spaced out. Might as well have been at the next page. Not that it matters. But this is pretty much, and I'm gonna stop right there. Get the book. I ain't gonna pretty too much tell y'all much more of that, but get the book. So I'm gonna put the link in the chat again. My book argues the case, Shazmo. If you ain't got it, get the book. Now, let's get a little bit more specific about this evidence against the veracity of SARS-CoV-2 genomes and intermediate between lineages A and B. All right, so I'm not going to go through too much of this, but I am going to give you the earlier onset, and then I'm going to get into the results. Just a little bit of results, not the whole thing, really the conclusion of it. All right, so early SARS-CoV-2 genomic, you can't see it. Now you can see it. Early SARS-2 genomic diversity can be separated into two primary lineages. You got lineage B includes the reference genome HU1 and is defined by nucleotide C87A2 and T28144, whereas lineage A defined as a substitution C87A2T and T28144C relative to the reference genome. Now, intermediate sequences containing either one of those, but not both, have been reported from earlier 2020. We refer to these genomes as CC and TT because they have the same nucleotide at these two key sites. And here we investigate the veracity of these sequences and concluded as probable that neither CC nor TT genomes circulated at the start of COVID-19 pandemic. They are likely the result of sequencing or bioinformatics issues. All right, so they get into the findings and deal with the data. It's a long conclusion and it goes through a whole bunch of stuff. I don't want to give it to you all, but I do want to give you the onset. This is important here. All right, this is important here. As discussed, the repeated occurrence of numerous derived mutations on either side of a given mutation is difficult to reconcile through homeoplasy events. Of the 77 mutations seen in CC intermediate genomes, 32 would need to be homoplacious if these CC intermediates actually existed. Similar 7 of these 12 mutations seen in TT genomes would need to be homeoplacious if the TT intermediates truly existed. These apparent homeoplacies can arise from issues regarding sample preparation, contamination, sequencing technology, and or consensus calling approaches. In particular, it seems likely that the nucleotide of the HU1 lineage B reference is frequently being called at these two sites. The findings cast substantial doubt on the veracity of CC and TT intermediate genomes in early 2020. We suggest that these earlier CC and TT genomes are erroneous and should be excluded from phylogenetic analysis. That is what one person argued. Now, I did that for a reason. Because there's a paper that gives more credence to both and goes from there. So we have to pay attention and let the science speak. We still have more data that is needed in order to track it, but we need epidemiologists on the job, and we have to have people to trust the science. And the way that you trust the science is to know how they are tracking these things. How they are tracking these things. What up, Aunt? What was Rand Paul hitting Dr. Fauci with? I ain't remember the term. It was basically it was about U.S. No, yeah, I had mentioned that already, Shazmu. In my book, I hate to keep referring to my book, but in my book, you had people that were claiming that it was gain of function. You're talking about gain of function, and really, what Rand Paul was arguing, when he was arguing from ignorance, and he pretty much was regurgitating other people. So his understanding of gain of function was inaccurate. If you're constituting gain of function, that means you're saying that this virus was created in the lab. And if you think that SARS-CoV-2 at the way the spike protein was facilitated was a lab generated, why come they can't replicate it? When they sequence the spike, when they sequence the virus, what they've known or what they've identified is they identified the place where, if this was manipulated, where they can pinpoint it on the virus where it could have been manipulated at, because they've tried to do that. And I keep telling y'all, the receptor binding domain is a significant part in this entire argument, and nobody want to listen to that. The spike is the spike, but the receptor binding domain is the key crucial piece of evidence here that it's natural. But we were Jave been SARS one. Retype that before. Retype what you're saying. But we were Shazmu. Have you been drinking? Is it not similar? No, it is. Heck no. SARS-CoV-2 is nothing like HIV. SARS-CoV-2 is, they're two totally different viruses. SARS-CoV, what kind of, all right, so you don't have the book, so you may not understand this at all, but anyone in the chat that's listening right now, what kind of virus is SARS-CoV-2? And what kind of virus is HIV? What kind of virus is SARS-CoV-2? And what kind of virus is HIV? Nah, bro. Shazmu, no. Them spike proteins ain't nothing alike, bro. Nothing. What up, brother Mathis? Again, ladies and gentlemen, what kind of virus is SARS-CoV-2? If you got my book, you know the answer. What kind of virus is SARS-CoV-2, and what kind of virus is HIV? That's an honest question, one that people who have my book should know. Don't go flicking through it, looking in chapter 1. Nah, Shazmu. Shazmu, quit guessing and get the book. Support the book, Shazmu. Hey, I do believe on this case right here, Shazmu, you're gonna appreciate it. But I'm gonna give you the answer, Shaz. So HIV is a DNA virus. HIV is a DNA virus. SARS-CoV-2 is an RNA virus. There are two different types of viruses. You have a DNA virus and you have an RNA virus. All right. So let's see if we can help you out. Let me see if I can pull up what the structure of a virus. Structure of HIV virus. Let me try to pull it up, and let's see if we can get some images. Shazmu. Let me share my screen. My bad. We can do this live. Appreciate you even asking that. It ain't a dumb question either, so don't trip on that. All right, so let's see if I could get you an actual structure of that virus. This is why you saying what it's saying about the spike and stuff like that, and people can really confuse it. All right, so let me see events. Let me see if Vince Rack and Yellow can do it. All right, so here we go. There's one, and then let's get SARS-CoV-2. If we look at them under a microscope, they look different, but here we go. Yeah, this will do right here. Open a new tab. So this is the structure of a virus, but this is a different one. It's influenza. I need the AIDS, so I went to the wrong one. I just use this. I don't like the way they look, bro. Because I can't get into what the answer the way that I need to. But do they act similar? No. No, they don't. We're not assisting in research on how early SARS and early similar bird and bat viruses and their transmission to humans. Not earlier on, but we did do some work. Now that I mean, I can't really answer the early part of that, Shizmu. I can't answer the earlier part of that. I know I kind of answered this already, so I really don't have to show you the difference. But once I slowed it down in my mind to do this, then I can actually pull it up and we can just look at the dang on viruses side by side. If this will allow me the ability to be able to pull up that image. And here it goes in the figure right here. So I'm gonna blow this up on the screen a little bit and make it big. And now we can look at both viruses. So you have a membrane protein, you see that. You don't see a membrane protein here. You see matrix proteins, but their matrix protein onset is green. Proteastases are these little things right here. Then you got your glycoproteins, which we see in both the S1 and S2, right? We see them here. That's our spikes. Then they got the glycoproteins here. Okay. And here. All right. We see the same thing there. We got that. And then we got that, right? Okay. Then you got your nuclear capsids, ssRNA bounds. You see this right here? That's that. Look at the capsids here in the proteins, circular. Then look at the reverse transcriptase. Look at the reverse transcriptase. And then the nuclear capsid bound RNA. See, these are indications that you're dealing with a what kind of virus? You don't have a lot of information. You don't have a lot of things going on inside of the middle of this particular virus. All the activity is on this outer shell. And this single-stranded RNA right here, I mean, why I say RNA? Nuclear capsid bound ssRNA is significant. It's together. This is split. Right. So now you know the difference between the right. So now we know we're dealing with an RNA virus here because it's single-stranded, and we're dealing with a DNA virus here because it's double-stranded. And that's how they rock. There's a lot more going on here, but this deals with the nucleus of the cell. This particular virus infects the nucleus of the cell. This particular virus does not infect the nucleus of the cell. Hold on, slow down, Shiz. I'm trying to catch up with you. So, because I want to go back to what you said earlier, where we're not assisting in the research. Oh yeah, I answered that. I really don't know in the earlier onset if we were assisting in it. I think we kind of came later, but they began to study bats and doing things like that because you have to remember Bill Gates. Bill Gates did some funding for the study of bats, similar like to what he did to Ebola. But I don't know about any other instances, so I can't really answer that wholeheartedly. Brother Mathis asked, he says, they was able to stop the HIV virus from replicating in the bloodstream. They can't stop the RNA virus. Is that right? No, Brother Mathis, that's not right. So what is correct is they can okay, they know how to pretty much deal with RNA viruses better because of mRNA technology today. But with DNA viruses, or specifically HIV, right? HIV replicates at a higher pace. So if you're infected with HIV, HIV has the ability to replicate inside of you. HIV has the ability to hide from your immune system. HIV also attacks your immune system. So whenever you try to create some type of defense against HIV, HIV in returns destroys the cells that are the good cells, right? They're able to attack your body faster than your body is able to adapt to it and fight it. Now, let me add something to this because there's been 2 known cases of people recovering from HIV wholeheartedly, naturally, 2. And HIV's been out since the 80s. And we need to see what the science is behind that, where these people have actually mounted a immune response and have fought off this virus. But nah, Brother Mathis, they haven't stopped HIV from replicating in the bloodstream. Shesmu says, but do they act similar? Well, in the example that I just gave to answering Brother Mathis's question about do the viruses act similar, no, because the coronavirus does not do that. It is not as elusive. And you may have heard Dr. Ko. If you watch Chill Talk or any interviews we've done with Dr. Ko, who works at Johns Hopkins, she specifically stated that RNA viruses are pretty much stupid. They're big. They're these big viruses. They're these viruses with these big spike proteins, and they really don't do much. The only way they can really truly introduce itself to your cells is through those spike proteins, which it latches on and does its due in that instance. But no, they're nothing like HIV. HIV is more elusive, more not transmitted, but that ain't the word that I'm looking for. It's more elusive, and it just mutates, and it does it live on throughout your body. All right, so Shesmu says, and how they attach. Okay, are they similar in how they attach to this? Well, yeah, well, all viruses attach pretty similarly. When they because they all have to be introduced to the cell. Just one just goes further than the cell introduction, and that would be the DNA viruses because they're gonna deal with the nucleus of the cell. So they all going to be introduced at that similar point, and then that virus is going to go further. Um, there's reports of new variant. Yes. Boom. I think that you was on the earlier show, but yeah, I brought out that information in the earlier show today when I was pretty much talking about how YouTube had, did what it did. And it's the I might get the numbers wrong, but it's sequenced as the B11529 or something like that. I might have it incorrect. Um, but let me give it to you because I got it. I just got to go back and correct myself. Oh, I was right. The B11529. Didn't I say that? I said B11529. Golly, that consuming things to memory. But yeah, the B11529 is 1.1529. That is the sequence of the new. It hasn't been given a name yet, or it hasn't been given a Greek letter yet. So it is a variant of concern. That's correct. Um, Shesmu says the human body is a powerful organism. Yeah, it's a powerful organism, but the human body is not the most powerful organism on this planet. Shesmu says, what was the so-called bat lady working on? Oh, good question. Let's go see, because I don't want to mess that up and be incorrect about that. So let's go see what the bat lady was working on. This lady right here. Remember her? We talked about her, but I think she was doing more than just that. But this is the bat lady that Shesmo is referring to. She was mentioned back in 2020. She even had some other information that she was working on. So Shesmu, you already see that she was working on SARS-CoV-2, but I think that you're referring to years ago. You're not talking about this year. You're talking about years ago because she received money for trying to find virus origins. All right. So Boone says flights from Africa into other continents are already getting banned. Facts. She is a super smart scientist. Absolutely. Yeah, this lady. Yeah, but look, COVID lab leak theory, China's bat researchers under scrutiny, you know, has no fear, says she did. Yeah, she handles her business. The bat lady is influential. We just gotta wait. But this is who she is right here, y'all. That's her name. Xi Zing Li. So during the COVID-19 pandemic, she and other people went out to research. Um, She even says the leading virologists have explained SARS-CoV-2 is most likely natural origin and that it is extremely unlikely that it leaked from a lab. She's colleagues, which studies emerging infectious disease, has noted estimates of 1 in 7 million people in Southwest Asia who live or work in the proximity to the bats are infected each year with bat coronaviruses. Y'all didn't know that. So published in Science Mag, they did an interview with her, and she gave out some information. So She gave out some information there, and they begun to do their little research and everything like that. So that's what the bad lady is actually dealing with, Shesmu. All right, let me see. Oh, boom cell. Yeah, I think you mentioned it. Yeah. All right, Aaron. Aaron says it's pretty blurry. Aaron, if you go in the settings on your cell phone, because I had to do it on my cell phone to make sure that y'all could see. If you go in on your cell phone and hit the three dots, change your quality from auto to advanced, and change it to 720, it'll be clearer on your cell phone. Chesmu says, I always thought that the body would somehow find a way to fight most, if not all, diseases. Well, the body ain't found a way to beat viruses like that. Vaccines, and see, this is Shesmu. This is where understanding vaccines come into play, because look at how useful vaccines have been. They've been so effective in our society and our understanding and our learning and everything, that a lot of these eradicated viruses we don't even talk about no more. We don't talk about them. There's no conversation related to these viruses that have been eradicated, because the vaccines are working that well. Uh oh, you talking about years ago? Yeah, years ago she was on some coronavirus study years ago, but I think Ebola and other viruses that she may have been looking at that time. Do you think of our lifetime they will be able to cure any of these viruses? Be more specific. You can't cure a virus. You can cure a disease, but you can't cure a virus. You got to remember, the disease is what people are trying to find a cure for. That means you've been infected with a virus, it crossed over to a disease. So let me give an example, Brother Mathis. So let's do it like this. You were infected with HIV. You developed AIDS. You have had AIDS for 15 years. And then comes an mRNA vaccine to fight HIV that prevents people from catching HIV, therefore stopping them from developing AIDS. And then there's medication that treats the disease. You see what I'm saying? So they'll have medications to treat a disease, but then they'll have vaccines that'll prevent the virus from infecting people. However, if they ever develop something that can prevent the spread of disease after people been infected with it, then that's instrumental. But that's just not how vaccines work. They don't work that way. Vaccines work to prevent disease, the spread of disease. If you already have the disease, then there's other treatments that will have to treat that disease or whatnot. And then, but yeah, I mean, maybe one day. Of course, it would be the detriment of the damn near the whole. Yeah. All right. Boom said exactly. Vaccines have worked so well. It's made people. Yep. Absolutely. Look. Boom. Boom. Hey. Facts. Facts. He says vaccines have worked so well, it's made people comfortable enough to become anti-vax. If measles, polio, etc. were still running rampant, nobody would be talking all that. Facts. That is an absolute big fact. And that is something I think, I don't mention it along the lines that you actually mentioned it in, but I mentioned it along the lines. That boy LeBaron's getting emails in Hawaii, boy. That boy LeBaron, he big time. Emails from Hawaii. But excuse me. Um, it just goes to show how well that they are working, that people can. But I like the way you said that people have become anti-vax. They work so well that it allowed people to be able to do that. But if you missed the beginning of the actual show, we began the show talking about a new vaccine. It's called Emergence Vaccines. And just to kind of sum up what they got going on, they're like peptide driven. It's a T cell vaccine. Its job is to target the T cell responses that will kill off the infected cells. And needless to say, the technology is new, of course. They are in the position to target specific viruses, viral diseases such as the dengue fever, Zika, Ebola, and the pandemic flu, and as well as serious intracellular bacteria infections. And what they want to do is deal with T cell priming vaccines. They'll have some intramuscular injection. They're trying to avoid needles. So there'll be a different design that'll be pretty much a needleless vaccine. Um, they'll deal with RNA and DNA, of course, and pretty much go from there. But if you want to catch that at the earlier onset of this particular bill, please do that. Cures are the best, but vaccines are needed. Very true. Very true. Vaccines prevent the spread of disease. They stop the spread of disease. Um, Let me see what else. But yeah, man, get the book shares, Moo. Quit doing all that clicking. Go on and hit that link. Boom. I don't know if you got the book either, but man, go get what SARS-CoV-2 taught me. I guarantee it. Just based on where you are in the conversation and what we're talking about right now, that both of y'all will really enjoy the book. I know that people will learn a lot from it. That's how the book was designed. It was designed to help us further the conversation and continue to keep things up in the sense to where we are illiterate enough to understand what the heck is going on around us with this so-called virus that is, man, uh this invisible as it is. It's invisible as it is, man. We're fighting a blind war. So we want to make sure that we're on the up and up, and we'll be able to manage to be in that fight and stay in it for the long haul. Let me see what's going on. But we don't really have too many people in here trying to argue today. I don't know if that's good or bad. Nah. Appreciate you, bro. Appreciate you. But yeah, man, if you get the book, of course, I'm going to sign the books. I got about 30 books. I've signed about everybody that's got a book from me directly. I've signed the books, and they got their books pretty fast. Yep, everybody that has gotten a book, and we make sure that we get those books out to people. Brother Marlon, I sent you an email 2 days ago about your book. So you got, yesterday. Yeah, yesterday about your book. Because you had got it that night. But I had you purchased it that night, and I made sure that I got you I got that book out to you ASAP. So your tracking number is in your email. And I appreciate that, my brother. You can ask Keenan. Keenan on the West Coast, man. Keenan to buy a book on Monday. His book be there on Wednesday. I don't know how that happened for him, man, but it be happening for him. Boom said, I'm outside the US. How do I get the books? Boom. Where you at outside the US? Go on the website, boom. If you go on the website, it says chat with us. Let me show you. This is what I need you to do. Go to the website or click on the link. You see where it says chat with us? If you will click this box right here, me and you, I have a conversation offline or off air, and I'll be able to answer anything that I need to answer for you regarding where you are. Okay, Australia. Yeah, yeah, I got you. Yeah, just hit me up and I'll be able to help you be able to get it the way you need to get it. I'll tell you how. Because I have some brothers. I have a few brothers in the UK who have the book. And that's a big deal for me that people in the UK have the book. Yep. It's just a fact. I got you, boom. Boom, way down at the land of what they call it down there in Australia, the land from up under or down under. I still ain't figured out how you get them books that fast though, Keenan. I ain't gonna lie to you, bro. I mean, when you know anybody that purchased the book, I put them in the mail ASAP, but I don't know how your books be coming the fastest, and you way out on the West Coast. That's crazy. So y'all, what I'm gonna be doing, and I know many of you are not. I don't know if you're on Facebook or not. As you can see, Pure Black is not in this chat. Pure Black is not in the chat. Because Pure Black is not in the chat, I don't know if he has Facebook, but my Facebook name is this for anybody. That's my Facebook name. I will be going live on Facebook to do refusions to misinformation videos. Facebook ain't taking them videos down. So if I put together something related to some he say, she say stuff, and I could source up and deal with it and get it on out the way, I prefer to do one whole video. And then what I think that I'm going to do is after I do the video, I may download the video and then I may clip it up to where I can edit it to where it deals, or somebody else could edit it to where it deals with the claim, like the person says plays the claim, and then the actual evidence comes up behind them and refutes it, and then go on to the next point like that. So people could probably link it to TikTok and Instagram and all of that. I don't know. I still don't know. I think that slows down what I want to try to do, but that is a creative way to refute some of these claims. I also ask you to get me into the sesh for real, Sean. We'll work on that. Oh, okay. You want to get into you want to deal with the sesh? You want to get into Ronnie Kemet? Huh? You want to deal with Egyptian spoken language? Or do you want to get into the script? You want to get into both, is what you want to do. You want to get into both. I'm gonna point you in the direction of Ujawu. You sign up for his classes and get you in there because I feel like he's solid in that area. And he a good teacher. He a good metal nature teacher. Well, Uncle, where you at? You somewhere celebrating Thanksgiving with your family, but then trying to act all black powerish? Where you at, Unc? All right, so I don't know what shares move at, but anybody else got any questions, comments, concerns, anything they want to be able to talk about, anything related to what we discussed earlier? For those of you who have the book, you got any questions? Are there, anybody with questions about the book? Yes, no, maybe so. No. All right, man. Look, we are hour 45 in. The Bills beating the Saints. Even after an interception, the Saints can't even move the ball. They got a whack quarterback. And then they just gave the dude $95 million. I don't know why. Number 7. And I ain't even seen him in the game. Taysom Hill or whatever. Dude his dude wasn't even looking for the ball. He was open right behind the safety, wasn't looking for the ball. Oh, let me put the link back in there too. My bad. It's in the link for the book is in the description of the video for the listening audience and those who plan a replay, but it's also in the chat as well. If you don't have the book, I suggest you get a copy of What SARS-CoV-2 Taught Me. You can learn so much from the actual book. And boom, we'll work it out on how to get you access to the actual book. And we'll lock it up on that for sure. I'll make sure you'll be able to get that copy. But if you know what? I'm gonna give it a few more minutes, man. See if anybody want to hop in. Bill, get some off your chest or whatever. Is iTunes sending out updates today? Or Max? They don't tell me to update my telephone number. I put the link in the chat for anybody that wanted to join and talk about anything related to what we just discussed today, or if you had something else that you wanted to build on. Okay, so it says Mac OS Monterey is out now. 12.01 to 12.13 gigabytes. You could upgrade to the new Mac OS Monterey. I'll do that when this show over. I keep my Mac updated. You're gonna catch me out here in these streets with some other stuff. It's a whole lot going on the well. Look, peep game, y'all. Kofi ain't said nothing. Oh, I know what it is. The Cowboys lost, so all the Dallas fans is mad. We ain't think about that. The dang Dallas fans is pissed off. But y'all big mad. Y'all lost by a field goal. Trash to the Raiders. The AFC West then blew y'all twice. Oh yeah, let me do this right quick. Shout out to my homeboy Avery, my best friend. Man, thank you for that reminder. Cause I straight up was about to not even pay any attention till I seen that notification. Let me go ahead and grab that right now. Where you at, Trap? There you go. Okay, so let me hit on your name. Let me go on and get that 2 right there. All right, I got it. Cool. Okay, Lido, Trip. All right, okay, cool. But yeah, man, I dropped the link in the chat, man, to see if y'all wanted to build. Anybody wanted to build? Okay, so I'm gonna go ahead and I'm gonna end this show, and then I may return. It's some people I want to attack. But yeah, I put the link to the book in the chat, man. Make sure you get your copy of the book. Come on, y'all. I need your help. Support the book. Get the book. Do your due diligence. Let's build on the book. On that note, I'm going to end this and I'm going to come right back. Different show. That's the way it go.